The cell cycle, or mitosis, is the stages a cell goes through in order to divide into two new cells. The cell cycle consists of interphase and mitosis. One somatic body cell that is diploid becomes two identical daughter cells that are also diploid, which means that they have a full set of chromosomes. Most of a cell's time is spent in interphase. In interphase, there are three stages. It's G1, S, and G2. In interphase, there are also checkpoints that check and make sure the cell cycle is occurring properly. Checkpoints are controlled by cyclins and cyclin-dependent protein kinases. Cyclins bind to CDKs to activate them. The purpose of a CDK is to tell that cell it can pass to the next stage of the cell cycle. CDKs work by binding to cyclins and creating a CDK-cyclin complex that signals the cell to proceed in the next stage of the cell cycle. When the complex degrades, the cell exits that particular phase. Growth and normal metabolic roles, which is synthesizing new proteins and organelles. There is a checkpoint during G1 called GO, or the resting stage. During this checkpoint, proteins check for cell size, DNA damage, nutrients, and growth. G1 cyclins bind to CDKs during this checkpoint, and the CDK tells the cell to exit G1 and move into the S phase. S phase is synthesis, where DNA is replicated so there is DNA for the new cell. G2 is short and the second growth phase. At the end of G2, there's a checkpoint right before mitosis. Mitotic cyclins bind to CDKs and create a mitosis-promoting factors, which is MPF. The MPF tells the cell to enter mitosis. This checkpoint also looks for cell size and DNA replication to ensure interphase went properly. The cell then enters the M phase, or mitosis. Mitosis has four stages: prophase, metaphase, anaphase, and telophase. Prophase is the longest stage. During prophase, chromosomes thicken and become visible. Centrioles move to opposite poles, and protein fibers begin to form the mitotic spindles that attach the centromere of the chromosome. There is then a checkpoint called the spindle assembly checkpoint, or the M checkpoint, that checks for the chromosome's attachment to the spindle. The nucleolus disappears and the nuclear envelope breaks down. Next is metaphase. Chromosomes line up on the metaphase plate in the middle of the cell with the help of spindle fibers that attach to the centromere and the centriole. Next is anaphase. Spindle fibers pull apart sister chromatids to become individual chromosomes. Chromosomes get pulled to opposite poles of the cells. Last is telophase. Chromosomes disperse and become no longer visible. Nucleolus and nuclear envelope reappear. In cytokinesis, a part of telophase, a cleavage furrow forms that pinches the cytoplasm in the center, forming two cells. How are you going to keep the phases in order? Here's a trick. Prophase is preparation for the cell to divide. Metaphase is meeting of the chromosomes in the center. Anaphase is pulling the chromosomes apart. And lastly, telophase is the tearing of the cell into two identical daughter cells.